Hello and welcome to D32 Inheritance. This is both SL and HL. There is a lot of standards in this one, so we're going to go through it. There are 15 SL standards and then 6 HL standards. So inheritance is really just a fancy way of saying we're going to be talking about genetics. This is what you're probably traditionally used to it being called. It starts with our first standard is saying, well, how do we do inheritance? How does it work? So this is, we take haploid gametes and we, from parents, and fuse them to get a diploid zygote. This happens in all eukaryotes that have a sexual life cycle. So not all eukaryotes, but all with a sexual life cycle, they do this. There are some that have half their life is sexual and half is asexual, like fungi, and so a lot of fungi. And so what they do is they only do it for part of the time. So this all comes back to that meiosis that we learned about, where you form gametes. And so a gamete, remember, is another word for a sex cell. And it's typically N, whereas a diploid cell is going to be represented by 2N, where N refers to the number of chromosomes. So N is one set, 2N is two sets of chromosomes. So you have two copies of each autosomal gene. Autosomal means not on the sex chromosomes. So humans have 22 pairs of autosomes and then one pair of sex chromosomes. All right, standard 2 is flowers. So for flowers, we do what is called a P generation. Then we cross those and we get an F1 generation. And then you cross those and you get an F2 generation. So in plants, we use P, F1, and F2 to represent them. So the pollen is the male part, and then the eggs are located in the ovary, and that's the female part of the plant. And so some plants can self-cross. So for example, pea plants, like Mendel studied, can self-cross. So they can self-fertilize because they have both male and female parts. But they often do not. And so how do we get new plants is by crossbreeding them or taking different varieties and crossing them to get new patterns and new variations. So how do we get all those cool looking plants you see? Those are coming from a cross like this. All right, standard 3 is a definition. Genotype is the allele combination. And so it is what copies of the alleles you got. And so it's typically represented with letters. And so we have like AA, so this would be dominant recessive, or you could have A and another letter, right? So if it's got multiple alleles, you could have that. And so if we have these are our 3 traditional ones for simple ones, would be these ones here. This would be heterozygous. And these two here would be homozygous. Homozygous. Okay. So they're combinations of the alleles. Now notice, the gene is both letters. The alleles are one letter. So that's an allele. Allele versus a gene. You have 2 copies of every allele because your cells are diploid. That goes back to standard 1. Okay. Standard 4. Well, standard 4. Phenotype. What is phenotype? So the definition of phenotype is the observable trait. Now, this is where it gets a little complicated, is it is based off your alleles and the environment. So we learned about gene expression sum, where you can turn certain traits on and off. That is what we're seeing here. Standard 5. It's not standard 5. Standard 5. What does standard 5 say? Standard 5 says that you have both dominant alleles and recessive alleles. And so a dominant allele masks the recessive allele, right? Like overpowers the recessive allele. So the recessive allele is only seen when homozygous. Now, not all traits work like this, right? Only our simple dominant traits work like this. But what you get from this is that there are phenotypes that only show up when you have both recessive alleles. This does not make them more rare. It just means that you'll only see them when they're recessive. So for example, having 5 fingers is a recessive trait, and then not having Huntington's disease is a recessive trait. And so 6 fingers and Huntington's disease are not that common, and so even though they are dominant alleles, they're still not that common. So do not mistake recessive for rare. Okay, recessive just means can only be seen when homozygous. And if it's advantageous, it would show up more often. It's hard to remove dominant alleles from the population completely, especially with large population sizes. We'll talk about that more with Hardy-Weinberg. Okay. 6. Phenotypic plasticity. What is phenotypic plasticity? This is the capacity to develop traits for the environment. Now notice, this is not heritable. Okay, well, phenotypic plasticity is heritable, but the changes aren't necessarily heritable, right? So this is something that is changing, and the classic example is the rabbit with the fur that goes and it gets darker in the cold because it needs to absorb the heat so that it doesn't, like, it's darker in the cold, especially in the extremities, so that those don't freeze or get frostbite. And so what it does is it goes back and forth, triggered by the environment. Okay. This does relate to gene expression if you're in HL. It does relate to that concept, but this is more of a like signaling, chemical signaling, which affects the gene expression so that it can go back and forth. Okay. All right, 7 is our first example we need to know. It's about PKU, which is a simple recessive allele, and so it's a simple dominant gene, and PKU specifically is the recessive allele. So it's Mendelian inheritance of simple dominance, but it's the recessive allele, and so it will only show up when it's little a, and it prevents breakdown of phenylalanine. So phenylalanine is an amino acid that you get in your food, which is normally converted to tyrosine. But if you have PKU, you can't break it down. This leads to the buildup of the amino acid, which can cause neurodevelopmental delays and can lead to a lot of problems. And so when you look at food, it will often say like contains PKU, so that people or parents know to not give it to their child who has phenylketonuria. So it's a recessive allele, so it would only show up if both parents had a copy of it, and it would be this box here. Or if one parent was recessive and the other was heterozygous, then you could end up with PKU. SNPs are something we've talked about multiple times. Now, for this one and a lot of the other ones we talk about, they're pretty simple, but we think about it like, oh, there's this allele and then there's this allele, but there are an infinite number of possible alleles for each gene. All it takes to make them is a mutation. And the most common mutation that doesn't break the gene completely is a single nucleotide polymorphism. This is where a single base is changed, and then that changes an amino acid. So you change a DNA base, which changes the mRNA, which changes the tRNA, which changes the amino acid. Okay? So that's just how it works if you've forgotten. And so what happens is you can get more alleles. Every mutation that is not completely harmless and is not removed from the population creates a new potential allele. But the change has to do something. We learned mutation rates are pretty low. So yes, technically you could have infinite number of alleles, but it's normally pretty rare because of natural selection, and mutations are deleterious, which means they don't stay around in the population normally. Now, this is not saying you have more than 2 copies. It's saying that you could have like an AC or like a BE. You could have any different like combinations of alleles, but you still only have 2, because you still only have 2 copies of each gene. Okay. Why did we talk about this? Well, it actually has happened and been pretty common for a lot of genes. The one we're going to focus on is blood type. So blood type is our example of the multiple alleles we just talked about. And so you have 3. You have your A allele, your B allele, and your O allele. Okay? So these are your alleles. You would have 2 copies of each of these, and you get your different blood types. This one here are codominant, and they're both dominant over I. Hence the reason it's sent like this. And so what you see, you have codominance and then simple dominance with the other ones. And so if A and B are expressed at the same time, you would get both A and B. Once again, this refers to the antigens on the outside of your blood cells, whether you have the A markers, the B markers, or in the case of O, no markers, which is why it's the universal donor, which is why your buddy here got debated into giving blood again, finally. All right, now, we talked about this a little bit already, but that one was codominance, but there's actually 2 different kinds. There's incomplete dominance and codominance. And so codominance, we just talked about, is when both alleles are expressed. And you get discrete traits, or a dual phenotype. So you see both, a dual phenotype, or like 2 discrete traits. In this one, you get a blending of traits. And you are supposed to know specifically about the 4 o'clock flower. Yay, 4 o'clock flower. It's also known as the marvel of Peru. Marvel of Peru, or the 4 o'clock flower. They're the same thing. We love that. The full name, or the species name, is Mirabilis jalapa. I don't know why I told you that. And so this one is the colors, okay? And so these are the colors of the flowers. I can never remember what it is. Yeah, it's the yellow, yellow, what? There are new yellow ones. Remember how we talked about how you can breed flowers and make weird ones? The traditional marvel of Peru, though, are white, pink, or red, where their genotypes look like this. And since it's incomplete dominance, there is no such thing as a dominant allele anymore. The little a, even though it is a little a, is not actually recessive, and so actually I probably should have done it like this. That's probably a more accurate way of describing how it works. Although people get really nitpicky about this, and I don't think it really matters, especially not at the level we're at. All right, 11. Sex determination in humans is based off of the sex chromosomes. So for the sex chromosomes, you have 2 of them. You can either have an XX or an XY, or you can have Turner syndrome where you have just one X, or Klinefelter syndrome where you have 2 X's and a Y. There's a variety of other chromosomal disorders you can have. So it's not actually quite as simple as this, but it's, for all intents and purposes, we are going to focus on the XX and the XY. Now, for this one here, this has a gene called TDF, which is called the testes determining factor, which causes you to make testes. That is what determines your sex. You have a Y chromosome, you don't have a Y chromosome. If you don't have a Y chromosome, you don't have the TDF gene, you don't make testes, and you don't develop testosterone or a penis or into a biological male. Okay? Almost all the other genes that matter are on the X chromosome, right? And so there are genes that matter on this one for both biological males and biological females. And so you can see here, males only have one copy. So if this is damaged, and this is why sex-linked disorders are more common in males than females, because they only have one copy. So if they have the broken copy, they are in trouble, which leads us to, there's two examples. The one you have to know is hemophilia, spelled the British way, annoying, with an extra A, hemophilia. Okay, that really makes no sense, whatever, it's fine. Hemophilia. Hemophilia is a blood clotting disorder. It's the inability to form blood clots. And so this is primarily affecting males. And so this is normal, it is recessive, and this is hemophilia. Hemophilia. And so what happens is females have two genotypes that are going to be normal blood clotting, and then only one that leads to hemophilia. So this is the only one that causes hemophilia. In males, though, you can see that if they had a mom who was a heterozygote, they would have a 50-50 shot of getting it. Now notice I said a mom who was a heterozygote, because one, males can never be heterozygotes for sex-linked traits, and two, the sons always, this is a terrible Punnett square, there we go, the sons always get the Y chromosome from their father, and so if you are a biological male and you have a sex-linked disorder, it was from your mother, right? Because see, this Y is right here, and then you can get, this is the interesting one, a 50-50 chance. If the mom has hemophilia, a son will always have it. Same with colorblindness, or at least the most common form of colorblindness. There are more than one form of colorblindness. Okay, that is hemophilia and sex-linked disorders. Colorblindness works the same way. It is recessive as well. Pedigree charts. Can I squeeze this on here? I'm going to do it. All right, 13 pedigree charts. Now, in pedigree charts, you're looking for what are called informative crosses, and these are ones that tell you something. Okay. This is an informative cross. This is also an informative cross. This tells you nothing. Even if this okay, this still tells you nothing, right? It has to be that like this, where and then something different happens. That's what makes the pedigree informative. And so for this one here, we know it must be recessive based off of this, because neither has it, this one does. And so the informative cross always looks like this. It's when you take two heterozygotes together and cross them, and you get the recessive phenotype. In this case here, it's dominant, and this is the recessive. So being normal is the recessive. So this one would be for a dominant disorder like Huntington's disease. And so you can see here, whatever skipped the generation is the recessive trait. In this pedigree, being normal was the recessive trait. So it is what you can see. Okay? So that is how that works. All right, that is hemophilia. Oh, no, that was 13. That's pedigrees. Sorry. Trick myself. 14. Okay. We have different types of traits. You have discrete traits where you either have one or the other, or sometimes the discrete trait can have 3 variations, right? We think of hair, and it's not actually this simple, but we think of hair as being straight, curly, or straight, wavy, or curly, right? 3 discrete variations. But the majority of our traits, we can see phenotypically look like this, and they're what are called continuous variation, and they can be represented with a bell curve or a box and whisker plot, which is what we'll look at in a second here. And so what you have is so like this could also, this one here, if I added a 4th column, this could also be our ABO blood types, right? So you could have this, if this was proportion of the population, this would be like A, B, and O. You can see how those are discrete. But this one would be more like height, where you have continuous variation. And for height, it's a combination of multiple factors. The first one being there are many genes that affect height. There's like 100 genes that affect height. And so each of them has a little like additive effect. And so you're like stacking up these boxes. And so it's here is like how many of your genes are on. And so if you have like all on genes, you would have this height. If you've got like half of your genes for tall are here, you'd be in the middle of the height. And so this one would be like, oh, all of them are on. And so you'd be really tall. But that's a low percentage of the population. The other thing that can affect continuous variation is the environment. So we know that the environment affects our traits, and the environment also gives us this this continuous variation. And so you can see we have our mean, median, and mode with this kind of data. You can also do a box and whisker plot. So a box and whisker plot is like this. You have your median, you have your first quartile, you have your third quartile, then you have your whiskers, and you have your outliers represented with dots on the outside, outliers. Your outliers are 1.5 times the interquartile range, which is 3q minus 1q. Okay, we calculate these quartiles by doing your traditional like 1, 2, 3, 4, 5. Look 2, and so for this, the median would be 5.5, and then you would repeat again. If this is the median, like let's say I had an odd number, if this had been the median, you do not use it for the first quartile. Minimum, max, oh sorry, this is our max, and this is our minimum. I was like, I didn't label something. The whiskers need their labels. 1, 2, 3, 4, 5, 6. Great, that's the 6 things you need to know about box and whisker plots. That leads us to the end of the SL, so now we have our HL only. So our HL only is starting with, well, how do we get different allele combinations? Well, that has to do with independent assortment. Of alleles. And so alleles for different genes assort independently if they're on different chromosomes. And this goes back to meiosis. And this is the random orientation. Yeah. Random orientation during prophase 1 causes them to sort separately, and these are called unlinked genes. Unlinked genes. And so you cross them, and so if you do what's called a dihybrid cross, which is where you consider 2 genes at the same time, you cross them, so you'd have like gene A and gene B, and you cross them. This was a terrible one because it doesn't tell you. Look, little b, and then they would look like this. I don't know why I chose double homozygous. That was not very good, and doesn't help us with the next one. Oh well, here we go, next one. So, why do we care about that? Well, we care about that because sometimes we're like, well, why does... Often we care about it when it's the opposite is happening. Like, wait, why does that fly always have white eyes and something else wrong with it? And that's because of linked genes, which we'll come to in a second here. Now, 17 says, well, Punnett squares. There are 2 you need to be able to do. really well, and that's a double heterozygous crossed with another heterozygous, and then a heterozygous crossed with what's called a test cross or a double recessive. Our recessives, of course, are going to be homozygous. Homozygous, that's the definition of recessive. And crossing something with a recessive is called a test cross because it allows you in the plants to tell whether it's homozygous dominant, the other one, or heterozygous, right? And so the one we care about is heterozygous, right? So that's a heterozygous. The results would be different. We saw what homozygous, that was worthwhile. Good work, me. We saw what homozygous dominant would look like as a cross. It'd be really boring. They would all be dominant for both traits, right? Dominant, dominant. But that will not be true if it was heterozygous. So what you do for a dihybrid cross, right, is you have to make, for this one here, you figure out, okay, how many different gametes can I form? Well, you can do a big A, big B, because they independently assort. You can do big A, little b. You can do a little a, big B, and a, little b. So you get 4 different gametes, so you need 4 columns, and then since it's the same, you also need 4 rows. 2, 3, and a terrible 4th row. There we go, got it. Okay, and then we'll do it the same, not that it matters. Okay, and then you fill out the box, right? Little a, little b, I'm going to kind of do this. And this one is recessive. It is the only one that will be recessive for both traits. Okay. When we do dihybrid crosses, we pretty much only care about the phenotype ratios. And so we have dominant for both, dominant for one, dominant for other, and then recessive for both are what we have here. And so we can see we have one box that is recessive for both. We have, did it, good. 9 that are recessive, or dominant for both. 9 that are dominant for both, 1 that's recessive. And then we have 6 boxes left, and what we can see is we have big A, little b, so it's recessive for this one. Big A, little b. This one is also recessive for the b's, and so is this one. So there's 3 that are recessive for B, and then this one, and this one are recessive for the A's only. So 3 dominant for B, recessive for A, 3 dominant for B, or A, recessive for B, and then that. So that's called the golden ratio, 9 to 3 to 3 to 1. It is worth memorizing that ratio. The other Punnett square, which is way easier, for this one, the only thing we can choose is little a, little b. We go like this. Notice that it's always a different letter, right? It can't be big A, little a, because that would be choosing 2 copies of the same gene, and that's a no-no, okay, right? That doesn't happen. For this one here, we can see, okay, this one's recessive for both, recessive for A, recessive for B, recessive for neither. So you get a 1 to 1 to 1 to 1 ratio, okay? So a heterozygous cross recessive gives us 1 to 1 to 1 to 1. A heterozygote crosses a heterozygote gives you a 9 to 3 to 3 to 1. And that will always be true. All right, so well, why does this happen? It has to do with like loci. So you have your chromosomes, and they have what are called loci, or the locations where they're found. And so each one will then create a protein. And if they're really close together, what we're going to see is those 2 proteins will always be pretty similar. And so this can happen in any animal, but it happens in humans as well, where it's like, oh, they're close together, so you see similar genes. That leads us to gene linkage. This only happens with autosomal chromosomes, right? Because there where we can see it. And so what happens is, instead of you having like this, where you could make combinations, you see A and big A, big A, big A, big B, little B are always assorted together. And so then what happens is, if we were to cross it like this, instead of seeing a 1-to-1-to-1 ratio, you see a roughly like 10, it depends on the linkage, but like 100 to 1 to 1 to 100. And those dominant for one, recessive for the other phenotypes in the middle are rare because these two are locked together and these two are locked together. And so you're supposed to represent it. Oh wait, this one, it wants you to use vertical alleles. No, of course it does. It's all right. Use the vertical, not the horizontal. That's my bad. It wants you to denote it like this. And so you can see how they're linked together on the chromosomes, so you can't. But there is an event called crossing over. And crossing over will cause you to get a new one where, oh look, now you do have this one because it crossed over during, oops, I said prophase 1 for random orientation. That should have been metaphase 1. Metaphase 1. Sorry, random orientation is during metaphase 1. The thing that made me remember that is because crossing over is what's during prophase 1. So when the homologous chromosomes group, they can cross over like that and exchange genes. Okay. The last thing you can do is you can do chi-squared to test whether they're linked or not. And so if p is less than 0.05, it means they're linked because they should have assorted randomly. And so anything greater than that means it was random. But if it's less than that, it means it was not random, therefore they're linked. And so what you'd be looking for is a pattern like this instead of something like 25, 25, 25, 25, which would be what you would expect. So your expected values come from the Punnett square, your dihybrid cross Punnett square, and then your actual values would have to be given to you, and then you could be asked to calculate it from that. Okay. All right. That is inheritance.